Hi everyone, we are from Group 5. Today we are going to present about the industrial microbiology and its application. First of all, what is industrial microbiology? Industrial microbiology is a branch of microbiology which deals with study and use of various microorganisms to produce products such as vaccine, antibiotic, alcoholic product, or to carry out important chemical transformations on large scale basis. Here is a brief history of industrial microbiology. Industrial microbiology originated with alcoholic fermentation process such as beer and wine. Subsequently, microbial processes were developed to produce pharmaceutical, food additive, enzyme, and chemical. Currently, methods for gene manipulation have given rise to a new microbial products, most of them which are not naturally produced by microorganisms such as insulin. There are some characteristics of microorganisms used in industrial microorganisms. The first one is they must be able to grow in simple and inexpensive growth medium. This will help to reduce the cost of adding additional growth nutrients to the medium. The next one is the end products of the industrial microorganism should be non-toxic and easily harvested. The industrial microorganism should be able to grow rapidly in the growth medium and able to produce desired end product under a short period of time to avoid contamination. Besides that, the industrial microorganism should be amenable to genetic manipulation. This is usually achieved by introducing beneficial mutations that alters the genetic makeup of the microorganism for industrial importance. In addition, the industrial microorganism should be genetically and physiologically stable. The next one is the industrial microorganisms should have less demand for oxygen during the fermentation process. And the last one is the industrial microorganisms should be resistant to microbial killer which may affect their growth. This picture shows the microorganism growth curve. There are 4 phases. The first phase is the lag phase where the microorganism metabolically active but not dividing. The second phase is the log phase or the exponential phase. It's a time of exponential growth of microorganism. The third phase is the stationary phase where the growth reaches a plateau as the number of dying cells equals to the number of dividing cells. The last phase is the death phase, which is characterized by an exponential decrease in the number of living cells. Next, I'm going to present about the primary and secondary metabolites. Metabolites are the products of metabolism. So during the different phases of growth, microorganisms produce different metabolites. For primary metabolites, it's going to be produced at log phase, which is the exponential phase. The red line indicates the synthesis of primary metabolites. The dashed line indicates the synthesis of secondary metabolites, usually begins at the end of the log phase and stationary phase. There are some difference between the primary metabolites and the secondary metabolites. Another name of primary metabolites is central metabolite. It is because of primary metabolites are key players in supporting the growth and reproduction of microorganisms. Primary metabolites synthesize at log phase. However, not necessarily all microorganisms will produce secondary metabolites. Therefore, secondary metabolites are known as specialized metabolites, which is not involved in microbial growth and reproduction because they are synthesized during stationary phase, not log phase. But secondary metabolites are essential for ecological functions and other activities. Next, primary metabolites are produced in large quantities, but secondary metabolites are produced in small quantities. Besides that, primary metabolites are not species-specific and thus might be identical in some microorganisms, while secondary metabolites are species-specific and thus are different in various microorganisms. In addition, the primary metabolites might from the molecular structures of microorganisms, while the secondary metabolites are not part of the molecular structures of microorganisms. Next, primary metabolites are not active in the defense mechanism, whereas secondary metabolites are active against invaders and might be involved in defense mechanism. The example of primary metabolites include alcohol, amino acid, organic acid, proteins, enzyme, nucleotides, and etc. For the example of secondary metabolites include antibiotic, zinc, and pigment. Next, I will pass the presentations to my groupmate.
I'm Ricky, and I will continue with the large-scale fermentation. In pharmaceutical industry, antibiotic can be produced through large-scale fermentation, and I will further explain about it in the next slide. Whereas for the food industry, my friend Natswa will explain about it later. The raw material required in production of antibiotic is carbon, nitrogen, and trace element. From carbon sources, soy meal which containing lactose and glucose are used as the food sources for the microorganism. Next, the nitrogen sources such as ammonia salt is essential in the metabolic cycle of the microbe. Trace elements such as phosphorus, sulfur, and zinc are needed to help the proper growth of the antibiotic-producing organism. Anti-foaming agents such as silicone may be required to prevent foaming during the production. Next, I will continue with the manufacturing process of antibiotic. The first step is starting the culture. A starter culture is taken from previous cold store desired microorganism and is grown on the agar plate. A pure culture is isolated from the agar plate and transferred into a shaped flask which containing nutrient in order to obtain the desired suspension. Then the desired suspension is transferred into a seed tank which contains all the necessary nutrients for growth. The content in the seed tank is mixed continuously and air is also supplied. The content in the seed tank is transferred to a fermentation tank after 24 to 48 hours. The second step is fermentation. Fermenter provide a favorable environment for growth and excretion of antibiotic. The operating temperature of the fermenter is from 23 to 27 degrees Celsius. Moreover, acid or bases are added to control the pH of the fermentation tank. The third step in the manufacturing process is isolation and purification. The desired antibiotic will be produced after 3 to 5 days in the fermentation tank. The antibiotic can be isolated from the fermentation tank based on the type of antibiotic form. The water-soluble antibiotic can be isolated by ion exchange method, whereas oil-soluble antibiotic can be isolated by solvent exchange method. Apart from that, the quality of antibiotic form is maintained by sterilizing the equipment with steam and frequent check on the purity of the microbial culture. The final step is refining. The antibiotic produced can be converted into various forms such as ointment, gel, capsule, and intravenous syringe. The converted antibiotic is packaged before delivered to pharmacy and hospital. This slide illustrates the overall process flow diagram of manufacturing antibiotic. The desired suspension is first fed into the seed tank and then goes through a sterilizer, which is functioned to control the quality of antibiotic produced before entering a fermenter. Large scales of antibiotic is produced in the fermenter. The produced antibiotic are then passed through a filter before it is being recovered as a product. This is all for me. Thank you.
Food industry lactic acid. First of all, I want to talk about what is lactic acid. Okay, lactic acid is catabolic product of primary metabolism by microbes, also produced by many higher organisms, including men who produce the acid in the muscle during work. Lactic acid products are formed from carbohydrate fermentation that are derived from pyruvic acid via the EMP, PP, or ED pathways. Products such as ethanol, acetic acid, 2,3-butanediol, butanol acetone, lactic acid, and xylitol production can also be produced as byproducts. A lactic starter is a basic starter culture with widespread use in the dairy industry. For this, making of all kinds lactic acid production is essential and the lactic starter is employed for this purpose. The lactic acid bacteria LAB. There are formicates group non-spore forming rods or corky shaped. Example for formicates group is Corynebacterium, Lactococcus, Lactosphaera, and for the non-spore forming Winococcus, Paralactobacillus tetragenicoccus, rods, Enterococcus, Lactobacillus leuconostoc, and for the corky shape, Prediococcus, Streptococcus, Phocococcus. Next is Lex porphyrin and cytochromes, grow anaerobically but are not killed by oxygen, as in the case with many anaerobics. They obtain their energy from sugars and are found in environments where sugar is present. They have limited synthetic ability and hence are fastidious, requiring, when cultivated with the addition of amino acid, vitamins, and nucleotides. So next is lactic acid bacteria into the two major groups. The two major group is homofermentative group that produce lactic acid as the scale product of the fermentation of sugars, and the two is heterofermentative group. Besides lactic acid, also produce ethanol as well as CO2, uses the enzymes aldolase. The next, move to the use of lactic acid bacteria for industrial purpose. The desirable characteristic of lactic acid bacteria as industrial microorganisms include ability to rapidly and completely ferment cheap raw materials, minimal requirement of nitrogenous substances, produces high yields of the much preferred specific lactic acid, ability to grow under conditions of low pH and high temperature, and ability to produce low amount of cell mass as well as negligible amounts of other byproducts. Is a raw material, cheap, low levels of contaminants, rapid production rate, high yield, little or no byproduct formation, ability to ferment with little or no pre-treatment, and year-round availability. When refined materials are used, the cost for purification should be cheap. Starchy and cellulosic materials because they are cheap, abundant, and renewable, where molasses have been used for lactic acid production. Starchy materials, sweet sorghum, wheat, corn, cassava, potato, rice, bread, and barley. These materials have to be hydrolyzed into fermentable sugars before fermentation because they consist mainly of a 1,4 and a 1,6 linked glucose. This hydrolysis can be carried out simultaneously with fermentation. Cellulosic materials. This material consists mainly of B14-glucan and often contains xylene, arginine, galactane, and lignin that have previously atomed to produce lactic acid from pure cellulose through simultaneous saccharification and fermentation (SSF). Some industrial waste products such as whey and molasses are of interest for common substrates for lactic acid production. Is about problems of lactic acid bacteria in industries. While using the LAB lactic acid bacteria in the laboratories, there are several chances of getting the contaminations and other problems, such as attack by bacteriophage inhibition by penicillin and other antibiotics, undesirable strands, acid produced by the lactic starters introduce elasticity in the curd, a property desirable in the final qualities of cheese.
Hi, I'm Sarah, and now I'll be talking about the products in industrial microbiology. First, in the pharmaceutical industry, we have tetracycline, which is an antibiotic produced by the microorganism Streptomyces aureofaciens. One method of tetracycline production is through shaker flask fermentation. The process includes preparation of culture, inoculation, fermentation, and recovery. First, preparation of cultures. This is done on worksman agar, where the pH is kept constant. It is incubated at 30 degrees Celsius for 10 days. The selected isolates are stored in oil or lyophilized. Next, inoculation. The volume used is 50 ml of culture per 250 ml of medium in an Allen-Meyer flask. A rotary shaker was used at 200 rpm. The inoculum used is dry mycelial spore transfer, liquid mycelial suspension, or lyophile. Then, fermentation at a temperature of 30 degrees Celsius for 48 to 72 hours and a pH of 4.5 to 5. In the recovery step, the culture broth is filtered to remove the mycelium. It is then treated with N-butanol or methyl-lysobutyl ketone in an acidic or alkaline condition to extract the antibiotic. Next, it is absorbed to activated charcoal to remove other impurities. Finally, tetracycline is extracted and crystallized. Factors being controlled. The pH is kept at 4.5 to 5, the temperature at 30 degrees Celsius, and the duration of incubation is 4 to 6 days for optimization. In addition, the genus Streptomyces can also produce chlorotetracycline under slightly different conditions. To limit the production of chlorotetracycline, it can be done by the elimination of chloride ions from the media or the addition of antimetabolites such as inorganic bromine salts in the media to inhibit the incorporation of chlorine in the tetracycline molecule.
I will proceed to the next slide. My name is Farahani and I choose mozzarella cheese in the fermented milk category as the food industry product. This is an illustration of general cheese production which consists of 12 steps. Before that, I will explain starter culture and non-adjunct starter culture for mozzarella cheese. The starter culture consists of bacteria of Streptococcus thermophilus and Lactobacillus delbrueckii single subspecies. This starts the fermentation process by producing the lactic acid from lactose conversion. It will then lower the pH, which stops further bacterial growth. While non-adjunct starter culture is a mesophilic isopolysaccharide producing adjunct mixed culture consisting of Lactococcus lactis single subspecies lactis and Lactococcus lactis single subspecies cremori, which can enhance the flavor of the cheese. The first step is raw milk will be standardized to optimize the protein to fat ratio to create a cheese with a high yield. Second is pasteurization. The milk will be pasteurized at 71.5 degrees Celsius for 15 seconds. This is to reduce the number of spoilage organisms and improve the environment for the starter cultures to grow. Next is cooling of milk. The milk is cooled to 32 degrees Celsius, which is suitable for the growth of the starter cultures later. For the inoculation of starter culture and non-starter adjunct bacteria, these two cultures will be added to the milk and held at 32 degrees Celsius for 30 minutes to allow the ripening process. The fermentation will occur where the bacteria of the starter culture will grow, the pH will become lower, and the cheese will develop flavor. For coagulation process, rennet containing enzyme chymosin is added where it acts on the protein milk to form curd. The curd is not disturbed for 30 minutes to form a firm coagulum. After that, it is allowed to ferment until it reaches pH 6.4. The curd is then cut with cheese knife into small pieces and heated to 38 degrees Celsius. This step helps to separate the whey from the curd. In the draining step, the whey is drained from the vat and the curd will form a mat. Next is making the curd texture. The curd mats are cut into sections, piled on top of each other and flipped periodically. This is to help expel additional moisture, allowing the fermentation to continue until a pH of 5.1 to 5.5. It will also allow the mats to knit together and form a tighter matted structure and mill into smaller pieces. For salt or brine addition, the curd is formed into loaf and then the loaf are placed in the brine. Then the cheese is formed into blocks, where the salted curd pieces are placed in cheese molds and pressed into blocks. However, mozzarella cheese does not require an aging process because it is usually eaten fresh. Other cheese such as cheddar cheese is typically aged 2 to 3 months. Lastly, the cheese will be cut and packaged into blocks or it may be waxed. There are 4 factors being controlled in cheese productions. Firstly is the temperature where the cheese should be kept at 32 degrees Celsius for the growth of starter culture bacteria. Choosing suitable milk with the desired composition is also important to produce high quality cheese. Next, it is also essential to choose suitable microorganisms in starter culture in producing specific types of cheese. Lastly, pasteurization is controlled by the types of cheese, which affect the temperature and its duration. That's all from us. Thank you for listening.